Hello everyone, myself Himani, and today we are going to discuss about the process of translation. So, um, uh, translation is the process in which genetic information present in mRNA is used to synthesize protein on ribosomes. Translation basically occurs in cytoplasm in both eukaryotes and prokaryotes, and the various material required for the process of translation are ribosomes, amino acid tRNA synthetase enzyme, tRNA, mRNA, etc. So in this diagram you can see here that this is the rmRNA. Its 5' end is capped and 3' end is tailed, and it also contains some UTRs, untranslated regions. Translation basically doesn't begin from here. It begins from AUG. AUG is a start codon. It represents for amino acid methionine, and it codes for methionine. And now what happens in next step, smaller subunit of ribosomes came at the 5' end and attached to it. Then at 5' end on the AUG codon, our tRNA initiated tRNA came, and it's with its anticodon loop. In this anticodon loop there are the sequence of amino acid there are sequence of codon complementary to this AUG codon, and it also carries amino acid. So in next step, one thing I tell you, that ribosomes has 3 site: A site, P site and E site. At A site is basically for the entry of new tRNA. P site is the site where polypeptide is synthesized, and E site is the site where the tRNA exits. So in this step, now what happens when you this AUG codon is basically on P site. Here in above step, this is on P site, so it is basically on P site. Now what happened on A site, a new tRNA came and it binds to the codons on mRNA. tRNA basically has anticodons, and mRNA has codons, so these both codon and anticodon gets binds, and tRNA bring with itself a amino acid. So the amino acid on P site tRNA and amino acid on A site tRNA both forms a peptide bond, and this peptide bond is formed by rRNA. Okay, so in next step what happens now, this ribosomes moves forward in the direction 5' to 3' end. Now what happens, the AUG codon here on P site, now it gets shift to E site and exits, and our ribosomes move forward. So next GUC codon, it came on P site. Sorry, there is some issues of light. Now it shift to I hope you can see. So it shift to P site. Now what happens, wait a minute. Yeah, GUC codon shift on P side, and both, and the sorry, I forget to tell you that above at P site the methionine amino acid on P site, it is also transferred to A site. Okay, now when the CAG came on P site, it has both amino acids. Now a new tRNA came on A site and it also has amino acid. Now they form peptide bonds, and the amino acid on P site get transferred to A site. Now again it shifts forward, and this CAG, it came on E site, and UGC, these codons came on P site, and In this way, on P site, polypeptide chain is synthesized, and it gonna move forward, and one by one the tRNA exits from E site, and polypeptide is gonna synthesized on P site, and A site, here A site, new tRNA comes. New tRNA comes with the amino acid, they form a polypeptide bond. The amino acid gets shifted to A site. When it move forward, then the polypeptide amino acid came on the P site. Next, in the end, what happens when this ribosome reaches at the UAA stop codon? tRNA doesn't bind with the A site. Stop codon signals for the termination of translation. Now what happens? A releasing factor came here and binds with A site. This releasing factor help in dissociation of the two subunits of ribosome. The polypeptide chain also get dissociated and released. So this is the whole process of translation. Thank you.